Hello there, all my students. I hope you're doing well amidst Corona crisis in our country. So, students, as I mentioned earlier, that I would be uploading soon the upcoming series of lectures on protein binding. So, here is the second one named Protein Binding 2 Types of Plasma Binding Proteins. As we have already discussed in the protein binding one about generally what is protein binding and what are the various types of protein binding, like the reversible or the irreversible one. And we discussed that as far as the pharmacokinetics is concerned, the reversible protein binding is of extreme concern. Here in this diagram, we can see that albumin protein, which is one of the important plasma proteins as its concentration is higher as compared to the other proteins. Now here you can see albumin is in combined form with the drug, the upper portion of the diagram, and through reversible process, the drug can unbind and can exist in the free form. Now, this free unbound fraction is what is important pharmacokinetically or as far as the pharmacodynamics is concerned, because only this portion could be distributed and can get available at the site of action. Downwards in the diagram, you can see as shown with the pointed arrows that bound drug cannot leave the bloodstream as albumin is unable to cross the small capillary pores. Now, coming to the topic of this lecture, and that is the types of plasma proteins. Plasma proteins are divided into three major groups, and those are first one is the albumin, second is the globulins, and third one is the fibrinogen. Now, the globulins are further divided as alpha 1 globulin, alpha 2 globulin, and the gamma globulins. If we look at the percentage proportions of the various proteins, you can see that albumin holds the highest proportion, that is 55.2% as compared to other proteins. Okay, now the albumin structure. Basically, it is synthesized in the liver and it is a single polypeptide chain of 6 10 amino acids. It is distributed in the plasma, in the extracellular fluids of the skin, muscles, and various other tissues, means it exists everywhere. Important amino acids which the albumin is composed of are lysine, leucine, valine, phenylalanine, and few others as given in the slide. Albumin got some important functions to play within the blood circulation, and normally albumin concentration is usually maintained at a relatively constant level, which is usually in the range of 3.5 to 5.5%, and this percentage is weight per volume basis. Albumin is also responsible for maintaining the osmotic pressure of the blood. Another important function of albumin is transport, as we know that the plasma proteins are involved in the transport of many endogenous and exogenous substances. So the endogenous substances which are transported by the albumins include bilirubin, cortisone, tryptophan, free fatty acids. Exogenous substances include the drugs. Drugs are usually weak acidic drugs which preferably bind to albumin through electrostatic and hydrophobic bonds. These are weaker bonds. The drugs include the salicylates, the phenylbutazone, and penicillins. These are highly bound to albumin. As I mentioned earlier, that albumin has a very important role in the maintenance of the osmotic pressure as well. Now, how it does that, as we can see in the diagram with red arrows, if you look at the right side of the diagram, albumin cannot cross the capillary pores easily. because of its larger size. And moreover, the sodium ions also get attached to the albumin very easily, and together they are able to retain water and build up the osmotic pressure in this way. Initially, while discussing the types of the binding proteins, when we discussed that these could be divided into three major types: the albumins, the globulins, and the fibrinogens. And globulins could be divided as the alpha 1, alpha 2, beta, and gamma globulins. And then alpha globulins can be further divided as the alpha 1 glycoproteins and the alpha lipoproteins. And alpha 1 glycoprotein and alpha lipoproteins are also very important proteins as far as the protein binding is concerned. My lecture is continued, and in the next series, I will discuss some other topics linked to the protein binding. Uh, I have given this link on this slide. I will provide it in the description box as well. It is a good website. You can go and visit it. You can find very useful information regarding the protein binding and the various types of proteins.